Okay, so I gave a talk last year in the summer school, so I wasn't sure how many people would still be here from repeating. Okay, so all of this will be new to you. So I called that talk Physics and Biology. What's the difference? And so this is going to be part of my theme today. I'm interested in the biology and the physics of a particular molecule called RNA, or ribonucleic acid. And for me, as you'll see, it encapsulates a lot of the special properties of biological systems. And I'll start out just by telling you a little bit about my personal history. I started out as a physicist. I did my bachelor's degree in physics, and then I started graduate study in physics at Purdue University many, many years ago. And as I was coming to my master's degree, I knew that I wanted to do a PhD, but I decided to go to the Career Center and register myself for jobs and just see what the job market was like. And I found that all of the people who were interested in interviewing me as a physics major wanted me for building weapons. And I didn't feel comfortable with that. So this was one of the reasons. I was already interested in biology. I was already interested in biophysics. But this was one of the motivations for me when I went to my PhD to shift to a PhD in biophysics. And I moved to Berkeley to do that. And I eventually did my thesis work and postdoctoral work on biophysics of RNA-protein interactions. And I was interested in it because it's physics, but it's physics within a biological system. So it has some special properties. And only because of that. So my idea of going into biophysics, vaguely I kind of thought, well, maybe instead of getting applications with weapons, I'll get applications in medicine that'll help people or something like that. But I really wasn't very interested in that stuff. That wasn't what was driving me. But as a postdoc at one stage, I was then working in Cambridge, England, in the Laboratory of Molecular Biology, and we were working on RNA protein interactions, and we were approached by some scientists from at least 2 drug companies. And they had some compounds that they saw as candidates for drugs against HIV. And they thought that those drugs might be acting at the level of the RNA. And there weren't very many people at that time working on the biophysics of RNA. So they came to us, and they asked us for help. And so then the people I was working with got this idea, let's start our own company. And they wanted me to go work with them. And so they started a company, and I worked for a little bit over 6 years. Thanks. And I worked for a little bit over 6 years in industry, in drug design, targeting RNA. Not because I went into my career trying to think about what's the application going to be. I was just interested in pure science, and I found myself in this situation where I was getting involved in biomedical applications. But the idea of targeting RNA for drug design was something very new, and it was very difficult. And you know, in industry, you have to get money very quickly. So our company, they decided, you know, there's a lot of science we need to understand with this RNA that we don't understand. So let's just go to some targets that people have already developed drugs against. And then we can do it very quickly. And I was still interested in the RNA. So that's why I decided to leave and go back into academia. And I wanted to go back to work on RNA again, to work on the pure science, but now with a conviction that what I was doing will have major applications in medicine in the future. How distant in the future really depends on how much interest and how much focus we're willing to put into the basic science in the RNA now. And as you'll see, I've looked at RNA as a biosensor and as a smart molecule. So smart molecule meaning that it has some properties that most of your ordinary physical systems that you'd come across in nature don't have. But it's something that's quite useful for a biological system. To be a biosensor, to be able to detect what's going on in the environment and then react to it. So these are properties that are essential for life. Now, since this is a physics class, I think we need to put everything into the context of physics. And I'm going to start with a very famous essay from Schrodinger. He gave this essay, you can find it as a little book, as a little leaflet, but he actually gave it originally as a lecture in the early 1940s, at the time of the war. And he was one of a number of physicists who were disillusioned with what was being done with their science, the destruction that was being wreaked around the world with their science. And so they started to get interested in biology because they thought maybe if we go into biology, our discoveries can be used in a more peaceful and beneficial way. And actually, it was people like Schrodinger, and in particular essays like this one, What is Life?, that actually led to the beginning of modern molecular biology. Modern molecular biology wasn't invented by biologists. It was invented by physicists, chemists, physical scientists, engineers, actually. And Schrodinger was one of the inspirations for it. So Schrodinger wanted to ask this question first, as a physicist, what is life? So from the point of view of physics, what makes a system living? What are the physical principles that allows it to operate in that way? And he started out saying, as a physicist, my perspective is that physics starts out with atoms, or small molecules, that are behaving randomly. And order in physics arises from statistics when I start getting a large number of small molecules. And he noted that in physics, we take advantage of symmetry properties and we take advantage of periodicity in these systems that contain large numbers of small components in order to come up with simple physical descriptions of them. So he says in physics, we look at systems that have this periodicity and symmetry, and I'm not quite sure whether that's because that's a characteristic of non-living systems, or whether because as physicists, we just choose those systems because it's simpler for us to model them in a very mathematical way. But in any case, most of physics works with these kinds of systems. So for example, sodium chloride crystal, we have a very repetitive structure here. And now, just looking at living systems, they seem to be ordered. So for example, you and I, our bodies are at 37 degrees, and whether outside it's at 0 degrees or 50 degrees, we maintain ourselves at 37 degrees, and from the point of view of physics, we know that requires a high degree of order, right? And you have to maintain a low entropy in order to have this big temperature gradient between inside you and outside you. So living systems are obviously very highly ordered. And living things are much bigger than atoms. So he starts to see where some of this is taking us in terms of thinking about it from the point of view of physics. So just to emphasize the randomness in nature from the point of view of a physicist, Schrodinger gave several examples. One is Brownian motion and diffusion. It's interesting to remember at this point that Brownian motion was discovered by a biologist looking through a microscope and looking at these small particles moving around and thinking they must be living because they're moving around. And of course, it turned out that they're not living. It turned out that this motion is just random motion, thermal motion, which is present at the microscopic level in the physical world. So this was a big discovery in physics, which came from investigations in biology. But it illustrated this randomness at the microscopic level in nature. Second example is a torsion balance. If you take a very fine torsion balance, the most sensitive torsion balance you can get, then you have a problem if you really start using it. What's going to happen? If I take a torsion balance that's sensitive enough to pick up, let's say, a nanogram or a picogram weight. When you try to put your weight on the scale, it's going to pick up fluctuations, right? And we know that there's going to be random motion in the molecules in the room. And if you make your balance sensitive enough, it's going to pick up those random motions, and you're not going to be able to detect your very small weights. So this is another piece of evidence that at the microscopic level, when you have small numbers of molecules, they move about randomly. They're not ordered. So we know that in statistics, if we look at the fluctuations in physics, then the number of molecules, number of components of a system is connected with the size of the fluctuations. So if we have a system with, let's say, n components, then the average error, or the fluctuations, increase as the square root of n. But on the other hand, the signal will increase as the number of molecules, right? So if we take the signal to noise, we're taking the square root of n divided by n. So the signal to noise, the ratio of the noise to the signal is going as 1 over the square root of the number of molecules. So the more molecules we have, the less fluctuations we have. So in other words, from statistics, we get order. What at the level of individual molecules is random and disordered, at the level of large numbers of molecules is ordered. Okay, so to summarize Schrodinger's argument, living things are bigger than atoms, much bigger than atoms, and they're ordered. And because of what we know from statistical mechanics, order arises from large numbers of small molecules. So now, Schrodinger's expectation is, when I look at living systems, I'm going to find a lot of small molecules cooperating together to give me order. So now, it's the early 1940s, and the big issue in biology is, where is the genetic information in the cell? So Mendel had discovered that there are genes that hold information in cells that determine the characteristics of a living thing. So for example, the color of a plant, color of a flower, and of course, you could extend it to us, the color of our eyes or our hair, and all these things are determined by genes. And people were looking for the source of these genes, the source of this genetic information. And we didn't know it yet at the time Schrodinger gave this talk. But what we did know was that this genetic information was located in somewhere between 2 and 48 molecules in a cell, depending on the organism. Some organisms have more or fewer chromosomes, but never more than 48 molecules. So how could this be? This is not what we expected. حاجة غريبة جدا, right? So this is Schrodinger's first observation, that biological systems are creating order from small numbers of molecules. We haven't seen this yet in physics. So here's a direct quote from his essay. The most essential part of a living cell, the chromosome fiber. So the chromosome fiber being the source of the DNA, the source of the genetic instructions, may suitably be called an aperiodic crystal. So what does he mean by that? He said that the physical systems, they have periodicity. That means we have small components, large numbers of them, that all are similar, or at least they can form some periodicity that builds up units that are similar, and this builds up order. But in the biological system, this chromosome, it can't be periodic. It's aperiodic. So it's like a crystal, he suggests, but it's aperiodic. In physics, we've dealt hitherto only with periodic crystals. To a humble physicist's mind, these are very interesting and complicated objects. They constitute one of the most fascinating and complex material structures by which inanimate meaning non-living nature, puzzles his wits. Yet, compared with the aperiodic crystal, they are rather plain and dull. The difference in structure is of the same kind as that between an ordinary wallpaper in which the same pattern is repeated again and again in regular periodicity, and a masterpiece of embroidery, say a Raphael tapestry which shows no dull repetition, but an elaborate, coherent, meaningful design. So this is where I get the title for my talk. So an aperiodic crystal. So as I mentioned, this essay by Schrodinger was very influential. And it turned out that what he predicted about DNA wasn't 100% right, but this basic idea turned out to be very accurate. And it was about a little more than 10 years later when the structure of DNA was solved. And this slide just summarizes the structure of DNA. So how many of you guys have seen this before, the DNA double helix? Okay, so about half of you. Okay, so for some of you this will be review, and for the others this will be something new, but it's something relatively simple, so hopefully you'll be able to follow this. The DNA is, I think, not really aperiodic, but quasi-periodic. So this is the structure. You have a chain and you have individual, what we call subunits. Each of these subunits is made up of a phosphate. This blue represents a sugar. And then we have a base. And so these individual subunits are connected to each other. So there's a kind of periodicity, but the base is not the same. That's what introduces the aperiodicity. So we have 4 different bases, A, G, C, and T. Now, how do these store genetic information? Remember how genetic information goes from parent to child. It has to be transmitted accurately from the parent cell to the daughter cell. So this sequence contains the genetic information, and it has to be replicated for the next generation. How does that happen? It happens because I can take 2 of these DNA molecules. And these bases have a complementarity to each other. So A likes to form these hydrogen bonds and form what we call a pair, base pair, with T. And G likes to form a pair with C. So if I have one molecule that has a sequence AGTC, then a partner molecule, to fit it, its sequence is determined by what we call base pair complementarity. So the information in this strand gives us the information in that one. Now these 2 can separate. They can go to 2 children cells, and this strand will determine the sequence of its partner, and vice versa. And both daughter cells will have the same sequence, and the same sequence as the parents. And this is what the 3-dimensional structure of this DNA looks like. We have 2 strands here, and you see how they're wound around each other in a kind of helix. So this is called the DNA double helix. And I'm gonna use that phrase. So I'm going to write it up here so that you'll know when I use this phrase, then I'm referring to this structure here. Now, I'm actually going to be interested in RNA. RNA is ribonucleic acid. DNA is deoxyribonucleic acid. Actually, chemically, they're very similar. Deoxyribonucleic acid. And then we have ribonucleic acid. What's the difference? The difference is deoxy. So if you look on the sugar here, if I have an RNA, I just put one oxygen here, actually an oxygen with a hydrogen, and that gives me my RNA. So chemically, it's very similar to the DNA. Okay, so I think some of the things that we can already start to see are distinguishing our biological systems from our physical systems are, in biological systems, we tend to have heterogeneity. We don't have this repetitiveness, this periodicity, which we have, at least in those physical systems that we like to study. So this seems to be a necessary component. This is another subject too, which I think would be a subject for another talk, which is biological systems are maintained far from equilibrium. So these are what we call dissipative systems. We have a complexity here that is a very high degree of complexity. But what I want to focus on in this talk, because it's something that is manifest at the level of a single molecule, which in some ways anticipates a lot of the interest that we have these days in nanotechnology and materials technology. Nowadays, we try to design materials that can respond to the environment, biosensors, things like that. But this was something in the 1940s that was very strange, right? But actually, living systems, they have the ability to respond to the environment. And what we're going to see is that a single RNA molecule can respond to the environment and make a genetic decision. So now, to go back to the early history of molecular biology, one of the main achievements, very early on, around the time that DNA was discovered, was the central dogma of molecular biology. If you hate biology and you only want to know one thing about it, especially molecular biology, this is a good place to start. This is the central dogma. The central dogma is stated in a very simple way here. DNA, arrow RNA, arrow protein. That's it. That's the central dogma. Very simple. What does it mean? It means that DNA, as we said before, holds the genetic information. Okay? So DNA is the boss. DNA gives the orders. And what does it give the information for? It gives the instructions to make a protein. So this is what the DNA looks like. It's double helix that we saw before. And the protein looks like this. The protein is folding itself up in a very complex three-dimensional way. It doesn't have this nice quasi-periodic structure like the DNA. And every protein folds in its own way. And the protein does the work. So DNA is the boss, and the protein, each protein goes out and does its own job. So why do we need this middleman here, this RNA? Because that's what I'm interested in. And if you look at the central dogma, it doesn't seem to be very important. It looks like it's just a passive messenger. It's just taking the directions from the DNA, the instructions, and then it's using those instructions, reading those instructions to make the protein. So why do we really need the RNA? And I would suggest that the first hint comes if we look at the structure in 3 dimensions. Because if we look at the other molecules, there's a connection between their structure and their function. So the DNA we saw in the earlier slide, what's it for? What's the job of the DNA? To hold information and transmit information. So the structure has been designed to do that. The protein, every protein has its own structure because every protein has its own job. As an aside, this is one of the reasons why proteins attract a lot of attention in drug design, because if each protein is unique in the way it folds itself up, we can take advantage of that because we can design a unique drug to go and attack it based on that 3-dimensional structure. Okay, so then what about the RNA? Let's look at it in terms of the structure. So we have, remember, we have this base pairing in the DNA. G always with C, A always with T. And this means that the sequence in one strand of DNA determines the sequence in the other molecule of DNA. But now the RNA. The RNA is chemically very similar, has G, C. Actually, T is replaced in RNA with U, but U is very similar to T, as it turns out. Actually, this is a methyl group here, and it's just replaced by hydrogen in U. So otherwise, we have the same kind of base pairing going on in RNA. في حاجة مختلفة جدا. مهم. What's the big difference? It's single-stranded. It's one strand. So the DNA has its perfect complement. But the RNA doesn't have a complement. So if I take one G here, my G's here in black. I know you can't see this sequence, but it's color-coded. So wherever there's a black here, it's a G. And wherever there's a blue, it's a C. So any one of these G's, it has a choice of many different C's that it can pair with. Same with all the C's, same with all the A's, same with all the U's. So that means that the RNA, using the same function that the DNA uses to recognize, to store and transmit information, can use that same mechanism to fold itself into all kinds of 3-dimensional shapes. And if it can do that, so from the point of view of physics, that means we have a lot of different states of the system, right? And the energies of those systems sometimes might be quite similar to each other. So the RNA can fold in a lot of ways, and it can perform a lot of tasks, like the protein. So a good hint for that comes from the structure of this RNA. This RNA is what's called the ribosomal RNA. What's the ribosomal RNA? The ribosome is actually made up of about 2 to 3 very large RNAs and something like 20 to 40, or I think even more than that, proteins. So it's a big assembly. It's a big machine. It's a big machine. What's it for? What does it do? It makes proteins. So this is an important molecule because it's this molecule, not just in you and in me, but in a bacteria, in a plant, in any living organism, they all have this ribosome, which is RNA plus protein. It's making all of their proteins. So it's like it's making all of the workers that are going to go do all the job of the cell. Now, it's a big, complex thing. I mean, you can't figure out anything from what I've shown you here, right? Because you can't see, these are individual bases, they're A, C, G, U, you can't see the sequence. But this is a kind of schematic way of representing the way that it's folding. So where I have these ladder steps, you see like here and along here, I'm representing base pairing. So I have the base pairing like what we saw in the DNA structure. So I have short regions that form double helices, but then you see I have these places where I have what I call a terminal loop or an internal loop. These bases are not complementary to each other. I have these unpaired bases. I have these branch points. What are they doing? We don't know structurally what they look like. So this was a huge challenge in biophysics, actually. And so let me ask you, how would you determine what this thing looks like in 3 dimensions? What's the 3-dimensional structure? How would you figure that out? I mean, the answer is going to involve some physical techniques. I'll give you that hint. You guys are studying physics, so... Okay, X-ray diffraction, yes. Okay, or how about other methods? That's actually the answer, but there are other alternatives that you might... Sorry? Electron microscope. Electron microscope would not have the same resolution as X-ray diffraction, but it could give you the overall shape. That's right. Yes, simulation using computers, but I'm asking about an experimental way. So the other way, but it's very difficult for something this large, is with nuclear magnetic resonance. Okay, but the best way, in terms of getting highest resolution, is X-ray crystallography. So we take our X-ray beam, we have our crystallized molecule, and you guys remember the rule for Bragg diffraction, right? So if we imagine that this molecule, this RNA, is made up of a bunch of atoms, each of the atoms is a scatterer, and remember the Bragg condition tells us that there's a relationship between how close together these atoms are and how far apart the spots are, the diffracted spots on a film here. So in principle, if we look at the pattern of spots here, we can go back and we can figure out where all the atoms were placed. But if I have in this RNA, I have literally, plus the proteins, remember there's lots of proteins with it, I may have a couple 100,000 atoms, maybe 1 million atoms. So it's a big challenge, big technical challenge to go from this data. Actually, it's a big technical challenge just to get the ribosome crystallized. But then once there, to go back and get this data and come back and get the structure of the ribosome is a big technical challenge. So it was done by biologists making the ribosomes, chemists crystallizing them, and then physicists solving the structure. And this was completed in 2000. And this is what the 3-dimensional structure of the RNA looks like. So you see that this 2-dimensional representation, this is just a schematic representation that just shows the base pairing, only the base pairing, which is what we have in DNA. But you have this complex fold, which is a lot like a protein. So the people who solved this structure got the Nobel Prize in chemistry in 2009. But when we look at this complex structure, it should make us think that this thing can carry out complex functions. So not just storing and transmitting information, it should be able to do other things. And this is physics school, so I don't want to go into a lot of the biology, because I could go and tell you about all the things now that we know that RNA can do, all the functions it can perform. There's kind of a list of them. I mean, the first one was that it can transmit information. I mean, this was shown this time that DNA was discovered. But now we know it can be an enzyme. It can catalyze reactions. It can even replicate itself. And when the ribosome was crystallized, we learned that it's actually the RNA part of the ribosome that's carrying out this reaction of making the proteins. So every protein in every cell is made by an RNA. And when we started to see some of these properties of RNA, that it has the capability to do the same things DNA does, and it also has the capability to do the same things that proteins do, then people started to think, well, maybe in the very early stages, when life was originating, before we had living organisms that had all of these components, the first molecule to replicate itself and to start acting as a biological system could have been RNA, or a molecule very similar to RNA. So this is a kind of speculation that's called the RNA World Hypothesis. And it illustrates the very unique properties that RNA has, the sort of life-giving, self-replicating properties of RNA. And just very briefly, I want to just test your patience a bit with a little bit more biology. And this related to the Human Genome Project and the genomics projects. And just in a very simple, overall big picture way, I want to focus on one very startling message that came out of that. So in this central dogma, let me write it up here again because it's important. Remember, the central dogma said DNA, RNA, protein. So this central dogma says all the purpose of all this stuff is to make the protein. Now, if I take the genome that's been sequenced for bacteria, for a bacterium like E. coli, on this chart in red, I show the portion of the genome, the percent of the genome, that actually codes for protein. That is, that actually conforms to the central dogma. And for a bacteria, it's about 90%. So for a bacteria, it looks like this central dogma is about 90% true. But now if I go to yeast, this red region becomes a lot less. And if I look at a worm here, this is representation of genome of a worm. Actually, it's only a minority of the gene that makes proteins. If I go to a frog genome, it's even less. And if I go to, I actually don't know what musculus is. I guess it's some kind of mammal. But if I go to humans, then the percent of the genome that's translated into proteins is just between 2 and 5%. So now it seems like this early idea about the central dogma only applied in complex animals to a very small part of the gene. So this leads to the question, so in the human genome, this is just 2 to 5% is going to here. So what percent then, I ask the question, is going to here? Is becoming RNA. So there were efforts, this chart I show you is from 2005, there were efforts to try to investigate this question, the ENCODE project. And the ENCODE project concluded that it seems in the human genome, at least in some cells, more than half of the gene is transcribed into RNA. So if we say something like 60% gets to here, and only 2 to 5% gets to here, then there's a huge part of the genome, whatever it's doing it as RNA. So RNA must be doing something else other than just coding for a protein. But the interesting thing is, when I say, let's say 60 to 93% of the genome may be transcribed, this is not all the time. And it's not in all the cells. Only sometimes in some cells. So this is something very different between the RNA and the DNA. The DNA, remember we said, it's transmitting information to the next generation. So it lasts in the cell for the lifetime of the cell. And then it's passed on to the next generation. But RNA is only present for a short amount of time. Okay, so now, again, I could go into a lot of biology and talk about, in the last 10 years or so, the new functions that we've started to learn that RNA carries out. I'm not going to go into that detail because it would be a lot of biology, but just what I'll summarize something about them that they all have in common. And that's that the RNA, even the presence of the RNA, is not universal for all the cells. It's only being expressed in some cells some of the time. Why is that important? When I was in graduate school in the 80s, I had no idea how it was that a cell in my eye, or a cell in your liver, or in your skin, all of them have the same DNA. Exactly the same sequence of the DNA. But they're showing completely different characteristics. How could that be? Now we're starting to understand that a big part of the answer to that comes in the RNA. So the RNA is the way that the gene copes with problems like this. Like your grandparents didn't have to learn how to type on a computer. Right? Okay? And maybe your grandchildren won't have to, because maybe they'll have devices where they just touch everything. Okay? Or maybe they'll have devices where they can just think and then they get what they want. Okay? So the cell's world is not the same world as its parent, and it's not the same world as its children. And not only that, it's changing within their lifetime. So it will have to change its behavior. So it has to sense what's going on, and then it has to adjust. And this is something that is very essential to life, and that distinguishes living systems from non-living systems. And RNA is a big component of being able to do that. So RNA, it's smart in that sense. It's helping the cell to make decisions on the fly. The DNA is the boss. The DNA lays down the law. Within that law, the RNA has to decide how it's going to be applied in the circumstance that the cell lives in. So now, this really sets up then the research part of the talk, which will be the last part of the talk. And I'm going to talk about a model system. You know, when you're an experimentalist and you want to understand a basic concept, then you need to find a specific system that can illustrate that concept for you. And the model system that I found very suitable for asking this question about how RNA can make these decisions. I mean, this is what I find interesting about this molecule. It makes decisions. It senses the state of the cell, and then it makes decisions. And the riboswitches are an excellent model system. What is a riboswitch? I will explain it through an example. Essentially, we can think of it as a mechanism for feedback control of the metabolic state of the cell. So I have a gene, and I have an RNA made from this gene. And the RNA is made, it starts out at one end. We call it the 5' end. I won't talk about what this means chemically. This means something very specific chemically. But RNA molecules and DNA molecules, they have a polarity. They have a 5' end and a 3' end. And the 5' end is the one that's synthesized first. So this 5' end is synthesized, and the riboswitch sits in the 5' end. It's the first part of the RNA to be made. And then it folds up in a very specific three-dimensional structure. And then the rest of the RNA that codes for the genes is made, and it sits downstream from it. And these genes that are downstream code for proteins, And the proteins are involved in biosynthesis. By biosynthesis, I mean it's just the cell is making some components that it needs. And in the case of this particular riboswitch, these biosynthetic proteins make this molecule here, which is called S-adenosylmethionine. You can't read it up here because the top of the slide is cut off. So I'm going to have to write it down for you. This is the molecule S-adenosylmethionine. And the rest of this talk, I'm just going to call it SAM. And I'll just briefly point out some of the chemical properties here. I have a sugar and I have a base. So this is actually quite similar, if you'll remember, to the subunit that makes up the DNA or the RNA. And then it has this moiety here is called a methionine, and it's actually an amino acid. So this S-adenosylmethionine, this SAM, it is created by these proteins that are downstream of the riboswitch. And when lots of this SAM is made, it comes back and it binds to the riboswitch. So this is where the biosensor comes in and the riboswitch. It detects the SAM. And when SAM binds to it, it changes its structure. So I'm representing the structure of the RNA here. There's actually two different structures depending on whether I have SAM. If I have SAM, I have the arrow go this way. Without SAM, the arrow goes this way. And where I have these latter steps, this is a schematic. Okay, so understand that I'm representing something like our double helical segments, remember, of the RNA. So in this structure here that forms when I have SAM, it's called the aptamer, I have 4 of these helices. So I name them P1, P2, P3, P4. It's just the 4 helices. And they come together and they form this structure. I call it the aptamer. And when this structure forms, then the biosynthesis stops. This turns off the proteins. So this is why this is a feedback regulation, because when the cell, the riboswitch, finds that there's SAM, it changes its structure and it says, you've got plenty of SAM, you don't need to make any more. So then we have a negative feedback which regulates the amount of SAM in the cell. So there are a number of reasons to be interested in riboswitches. So I'm interested in them, as I said, because they manifest these properties, physical properties that are very biological. That they sense the environment and then they make genetic decisions on the fly. But there are other reasons. It turns out they're mostly in bacteria. There haven't been very many found in human cells. And for that reason, there's interest in them as drug targets. So here, just by accident, again, I find myself in an area that has lots of applications in medicine. And also in nanoengineering. If you can imagine, this is a device that we could use to build synthetic networks. It's a very sensitive biosensor, actually. And here's just some examples. There have been a lot of riboswitches found now which react to a number of different molecules, and these are just some of the examples of molecules for which riboswitches have been found. And one thing that's interesting in light of this theory that I talked about, the origin of life, the origin of living molecules, is that many of these are molecules that resemble the nucleotide bases. This one is actually an amino acid. This is a component of a protein. So if we think about a world before proteins and before DNA, but that had RNA, RNA could find the precursors and gather them up. This is what we see with the riboswitches. So now, what do I want to do? I want to ask the question, how does this riboswitch work? How does it sense the ligand? And then how, once it senses the ligand, does it change this structure as we saw? Changes the structure from here to here. How does that happen? And the first step is to try to get the structure. And again, we can use X-ray crystallography. And this has been done by a group. First group to do it was in Colorado, the group of Rob Beatty. And this is the structure that they obtained by X-ray crystallography. So this is a representation of the sequence. It looks a little bit different than the representation I showed you before, but it has 4 helices, P1. Remember we had 4 helices, P1 and P4 are shown here in blue, and this is the 3-dimensional structure. Those 2 helices stack together, and then see how they make a continuous helix. And then green, we have the P2 and P3, and they form a continuous helix, but then you have a kink here. So you see how these helical regions, like you have in DNA, combine with some components where you have some kinks here and a little bend there, and it makes a very complex 3-dimensional fold. Very much like a protein that can give a unique function. The SAM is this yellow in here. So it finds a very unique 3-dimensional shape in the pocket, and it sits there. So this information then is very useful in terms of understanding the first function of the riboswitch, which is the biosensing. How does it find the SAM? But in order to do this, in order to get this X-ray structure, now we can't see the bottom of the slide, but this structure is actually part of a much larger riboswitch. And in order to do this X-ray structure, what these guys did is cut it right here. Okay? So it couldn't form this other structure. Okay? Because if you had the RNA, if it's jumping back and forth between 2 conformers, it's going to be very difficult to use the X-ray to pin it down in one and get the structure. So while this is valuable for understanding this component of the riboswitch that recognizes the SAM, it doesn't tell us how the switch takes place. So we cut the molecule here. We can't get the transition. But the function of the riboswitch is not just in the ligand recognition, and this is indicated very clearly in the literature, because if it was just the recognition of the ligand that determined the function of the riboswitch, then there would be a direct correlation between how tightly the ligand binds and how much of the ligand you need in order to get the response for the riboswitch. So let's think of it this way. If you have a ligand that binds very tightly, if there's a very big free energy drop associated with the binding, then with a very small concentration, very small amount of ligand, then the RNA will respond because the majority of it will be bound. But if you have a weak binding ligand, then you won't see a response until you get a lot of ligand. Okay? But this is not what we see in the cell, or even when we run the reaction in a test tube. We find that the threshold ligand level is much higher than would be predicted from the binding affinity. And also we don't see correlation between ligands that bind tightly and ligands that have a response at low concentration. So there's something else going on. And in order to understand that, we're going to have to look at other parts of the RNA other than just this aptamer that was used in the X-ray structure. And then I think one thing to notice in all this discussion is that this SAM molecule is much smaller than the RNA. SAM has a molecular weight of 400. The riboswitch RNA has, at a conservative estimate, even if you ignore the rest of the message, just the part that's the riboswitch, it's 40 kilodaltons. It's 100 times the molecular weight of this molecule. And yet this little pygmy of the SAM is rearranging the structure of this giant RNA. Okay, so how are we going to investigate how are you going to incorporate these other missing parts of the riboswitch and understand how the switch takes place? Well, one approach is to use some calculations using statistical mechanics. So now, remember we said that the RNA, each of these bases has a number of different choices of partners. So the RNA can fold in a lot of different ways with a lot of different combinations of partners. Each of these folds, we call them conformations. So the conformations if you just imagine any one of the A's, for example, it can pair with any U in the molecule. It can also just sit unpaired by itself. So there's a lot of conformations in an RNA that has, let's say, a couple hundred nucleotides. And each of these conformations has associated with it a free energy. And so we have a Boltzmann factor at any given temperature, which tells us the probability weight for each of these conformations. But remember, this is just a probability factor. I guess you guys you're taking some statistical mechanics. Have you come up with a partition function yet? Okay. So the partition function, of course, is when we sum all of these, and that gives us the normalization factor. Okay, so once we have the partition function, we can calculate the probabilities of these individual conformations. And from those probabilities, we could calculate properties that we can measure for the RNA. So for example, the base pair probability, that is the probability of any given one of these base pairs. This is something that at least approximately we can measure in the lab. So when I was at LSU, my student Wei Wang teamed up with Ju-Young Kim at the Center for Computation and Technology, and they did some of these calculations. And we published a paper in which, in addition to the calculations, Wei did a number of experiments in order to test the base pair probability predictions. And now what I'm going to show you the next few slides very quickly is just what some of the lowest free energy structures were. So the most highly populated. I'm going to choose the three most highly populated. And as I look at them, I want you to focus on 1G. We call this G11. So in the bound form of the RNA, this G is not base paired. So it's sitting in this gap between a helix here and a helix there. But now let's look at the structures that are predicted. So when we do the partition function calculations, we don't have the ligand. Okay? So this is a calculation of the probable structures in the absence of the ligand. So here's the first one. And I've highlighted the G and covering it up, but there's actually a C here that it's base paired with. And you see that this is within a more complex structure. I have here what I call the anti-terminator helix. This one prevents the formation of the P1 helix. So I'll just call it AT from now on. And this is what dominates in the absence of the ligand. So now here's a second conformation, and you see here's our G11 again, and now it's paired with a C, but it's a different C. So if you compare the two, you see different helices forming. And the G finds a different partner. Now here's a third conformation, and now we have the G pairing with a third partner. And now, this is the structure that was observed in the X-ray with SAM. And here's the G within that structure. It's not paired with anything. Okay? But when we do our calculations without SAM, then G11, this G, is predicted to pair with over 30 different partners. And that's by far more partners than any other residue in the molecule. So what our partition function calculations are telling us is that this RNA has a hotspot. This hotspot is able to go sample It's doing a sampling. It's sampling different partners. It's sampling different conformations for the RNA. And what the crystal structure shows is that SAM binds to this hotspot. So that's how this little tiny SAM molecule can rearrange the RNA. Because the RNA has set itself up to have a very small sensor, which is this G11. So we start to understand now how this can happen, how the small molecule can rearrange such a large molecule. And I'm not going to go into the experiments because that involves a lot of biology, but we found experimental evidence for at least some of the alternative structures that Wei predicted. And Wei did these experiments as well. Wei was an amazing guy. He does theory, he does computer simulations, he does experiments. And he called this SAM a small molecule with a strong arm, because the small molecule has this big effect on the big molecule. But it's actually not the strength of the small molecule, it's the cleverness of the large molecule to set up this sensor so that it can detect small things in the environment. So now, this tells us something how thermodynamically, let's say, the SAM can stabilize a given conformation and refold the RNA. But there's something else that comes into this when we look at this biology. Remember, we said the RNA doesn't sit around forever, right? And it also takes a certain amount of time for it to make the RNA. And if the RNA sits around for a long enough time, the ribosome will jump on it and start making protein. So the decision to detect the SAM and change the structure has to take place before all of the RNA is finished synthesizing. In fact, before the gene is synthesized. So it's important not just the thermodynamics, the thermodynamic equilibrium between these structures, but also the rate at which this folding is taking place. So I want to understand this process of switching. So I have two conformers, one of them with SAM. I form this helix here I call the P1, and the other one without SAM. I form this helix called the AT. And look, this red segment is a segment that changes partners. So I want to look more closely at this process, this switching. How does this partnership change? Well, I'm representing here the base pairing. And so we asked ourselves the question, could we have an intermediate in which my swapping strand is partly base paired with this helix here, partly base paired with that one? So in other words, so I have partly this red base pairing and partly this blue base pairing. So this is a kind of intermediate. So now we ask the question, would SAM be able to bind to this? Because we know that SAM binds to this one. It doesn't seem to bind to that one, right? But if I take something that's in between, will it bind? So we asked that question, and Vamsi Boyapati did some of these experiments with a technique called equilibrium dialysis. And the question is, how do you test that? How do you test whether the ligand can bind to the intermediate structure? Well, we can do like the guys did who did the X-ray structure. Remember, for the X-ray structure, they cut off this part of the RNA so that it could only form this aptamer. So what we did is we cut the RNA in different places. So we could force it to form a structure which is completely in the unbound form, or we could force it to form a structure, whether the ligand is there or not, that is a hybrid. And we could have different numbers of base pairs in this red helix and different numbers of base pairs in this blue helix. And so then he measured SAM binding to all of these intermediates, and he found that with the one here that doesn't have this red helix at all, we can't detect a binding. So this chart shows the KD. So the KD is the concentration of ligand at which the RNA is 50% bound. So the lower the KD, the tighter the binding. So high KD here for this P2AT, that means no binding. Very low KD here for the aptamer, that means very tight binding. And for these intermediates, what we found is an intermediate amount of binding. To me, this immediately suggests a mechanism for switching. So let's think about it in terms of our thermodynamics. Let's say I take a molecule like this one. So it has some helix here that's red, and it has some helix that's blue. And then it has some region where it can switch between the two. Now, if I plot the free energy without SAM, without the ligand, I know that the blue helix is favored. So if I plot the free energy against the conformation, and here I have the long blue helix. So I have 10 AT. AT is the blue helix here. That should be lower free energy than if I extend the red helix. The red helix is the one that forms with the ligand, but I don't have ligand now. So that must be higher free energy. Now I add the ligand. There's favorable free energy for binding to these guys, but not to those. And a little bit less favorable free energy for binding to the ones in between. So now my free energy landscape reverses. Now, the structures with the red helix become lower free energy. The structures with the blue helix are the same, but also the intermediate ones also lower free energy, and this facilitates my path to go more quickly. And Osama El-Aida, who's in the audience here, has been doing some calculations, or he was in the audience, I don't see him now. Yes, here's Osama, yes. So he's been doing some calculations. So he set up a thermodynamic cycle, and I can take the unbound, what he's calling here the unbound conformer, that's the one without SAM, that we normally expect to see without SAM. So in other words, without SAM, it's the one with the low free energy. And we can imagine the bound conformer without SAM, which we know is higher free energy, so there's a free energy difference between them. Now, if we add SAM, then the free energy of this bound conformer goes down relative to the free energy of the unbound. So we draw this thermodynamic cycle, and then we can do calculations. So we can repeat the calculations that Wei did without the ligands. We repeat them with the ligand. Now we have new free energies that take into account the binding free energy. So now here, I take all of my intermediate structures. So these were all the intermediate structures that have part of blue helix and part of red helix. And if I add them all together in the absence of ligand, then the sum of the probabilities of all of them is like 10 to the minus 4. But now if I add the ligand, the sum is about 1.5 times 10 to the minus 2. So that means about 1.5% of the time, it's about 1.5% probability of having one of these intermediate structures. That might not sound like a lot, but it's 100 times more likely than it is here. So I'm going to have a big acceleration of the rate, because once I form this structure, my ligand binds to it, and once it's bound, it's going to drive the system very quickly, because remember that free energy landscape, it's a downward slope to get to the aptamer. So now, finally, I'm going to end up showing you some movies with some simulations. So here's where we're going to apply some classical physics, some classical mechanics, in a very simple way, but combining it with computers. So what I want to do is I want to simulate this transition. I want to add the ligand, and I want to simulate the process of it converting from one structure to the other. So I use molecular dynamics. I start out my system. I kind of build an initial starting structure for the X-ray coordinates. So I have the X-ray coordinates from the RNA and also from the SAM. And I have to take into account, it's very important in biology, take into account the surrounding molecules. Water, ions, magnesium, sodium, etc. And I start out, these coordinates give me the positions So I have position vector associated with each atom. And then arbitrarily, I can give each of them an initial velocity. And then I can calculate during a period of time delta t how exactly the position is going to change. And then I know after the time delta t what the positions are and what the velocities are. And then I can start bringing in my physics. So I bring in my potential. And I can calculate the potential from the structure, from the coordinates. And the way that I do that is I assign a charge to the individual atoms. And so then I can have an electrostatic potential, and this is just a Coulomb potential. And then I model the bonds, the chemical bonds, as a mass on a spring, because you know they have a favored length. And we can model them as harmonic oscillators. And we do the same for the bond angles. And then we have other kinds of potentials associated with the hydrogen bonds and what we call the Leonard-Jones potential. The Leonard-Jones potential is related to dipoles, but not fixed dipoles, not fixed dipole-dipole interactions, but induced dipole interactions. And so we add together all of this potential, and we can calculate then the accelerations of each atom, and so then we get new velocities, and then this allows us, after a time delta t, to determine new coordinates. And we go through this step by step, one time frame delta t at a time. So it's not a direct analytical solution of the trajectory, because it's not continuous. It's a discrete simulation of the trajectory. Okay, so now what do we see? So I'm going to show you now a movie that Wei made. And we're going to start out okay, we start out with our AT helix. So the AT helix, I've switched colors on you now. This is the one that was blue before. Now it's red. This is the one that is favored in the absence of SAM. And then I have this blue one. This is the P1 helix is favored in the presence of SAM. And what I've done is I've taken an intermediate, because I already have some AT helix here. I already have some P1 helix here. But there's a region where the two can compete. So right now, when I begin, I have this red helix paired with the blue helix in this area. And this blue, I want you to watch this blue region here. It's sticking out on its own. But what we think is the SAM that's shown here, should influence the blue helix and give it the opportunity to come in and displace the red one. So let's see what happens. Okay, so you see the red helix is starting to get a little bit wobbly here. And then look, it's shoved out of the way, and the blue helix comes and takes over, just as we predicted. So now I'm going to show this again in close-up with the chemical structures. So just this region here, we'll blow it up. So now you see we started out with a nice helix here between this red and this blue, and this guy is sitting out on his own, and then jumps in, displaces the red strand. And now we slow down the simulation a bit, because I want you to see this moment when we get a perfect blue helix. And this happens, we repeated the simulation without SAM, and instead of the blue helix coming in and displacing the red helix, the little bit of blue helix we started out with was lost. So this transition seems to depend on having the SAM there. So how is the SAM doing it? How is the SAM facilitating this transition? Well, a hint comes if we look, here's the binding site of SAM, and we just zero in on the binding site, and we have this green sphere here which represents the magnesium. This magnesium is seen in the X-ray structure, and RNA is negatively charged. I have a negatively charged strand of RNA here, a negatively charged strand of RNA here, and in the X-ray structure, these two negative charges are close to each other. These two negatively charged strands are close to each other. So there has to be a way. SAM is positively charged, so it can help. Positively charged molecules have a lot of impact on RNA, because RNA is negatively charged. But SAM picks up with the magnesium, teams up with the magnesium, and becomes even more powerful. So we're setting up a team here, positively charged team, that's making a big complex that helps to stabilize that structure. And we can see this now if we just look at the simulation, zeroing in on this region. Here's the magnesium, here's the SAM. Here's with SAM, here's without SAM. And look at what happens to these two strands and what happens to the magnesium as we run through the simulation. So you see how they're very stable. These two strands stay close together. The magnesium stays in the same position. Now in the other simulation, I don't have SAM. See, magnesium is jumping around. And so I don't have the same kind of stabilization of this structure that's required for the formation of the abdomen. Okay, so I've pretty much taken up all my minutes, right? And no chance for questions. I don't know if we're going to have like 5 minutes for questions. But just to summarize the conclusions, what we've learned about how SAM refolds the riboswitch RNA, we learned that the RNA forms a hotspot. The ligand zeroes in on this hotspot, and that is how it manages to have a big effect. We've been able to simulate the strand exchange, and we've been able to come up with a model that explains how kinetically, not just thermodynamically, but kinetically, the ligand can facilitate the strand exchange. And we saw on the last slide that another key to this is electrostatic interaction with the SAM and the magnesium teaming up. And then if we take a step back and think about this more now in terms of RNA as this model molecule for biological molecules, what we've seen is that RNA has a unique combination of skills. Like DNA, it can store and transmit information, but like proteins, it can fold and form particular functions. And we've seen that these skills, this combination of skills, is particularly powerful in life for sensing the state of the cell and in helping the cell decide from the state of its environment what it needs to do to maintain itself in its structural form. So just very briefly, some of this work was done by my group at LSU. These were some of the students, and we collaborated with some of the people in the computing center there. And then also I want to acknowledge Osama for the work that he's been doing here just in the last few weeks on the partition function calculations. Okay, so and I thank you for listening, and I apologize I went over a little bit. We started a little bit late too, I guess.